Okay, Sarah, you and I are going to move here.
All righty. Hello, everybody. Welcome to this special session that we are having on LabArchives. Today we have Hannah Clark, who's with Enterprise Success in LabArchives. And we know there are quite a few of you who are going to be on today. So if you have any questions at any point during the session, please feel free to type up your question in the chat. We'll be stopping every so often just to answer your questions. If you want to be taken off the mic, just hit the raise hand button under the reactions tool in your toolbar at the bottom of your Zoom window. And we can take you off the mic if you have maybe a more in-depth question you want to ask. We're also recording today's session, so after we're done, I'll be sure to send a follow-up email with a link to the recording so you can go back and refer to it, as well as any helpful links that we might have during the session. So without further ado, Hannah, please take it away.
Perfect. Thanks so much. Hi, everyone. My name is Hannah Clark. I'm part of the LabArchives team. I'm joined here today by two of the site administrators for Weill Cornell Medicine, Michele and Sarah. So if you have questions about institution policy, I'm sure they'll be able to help out with that as well. I'll try to keep an eye on chat. I'll stop periodically as well. But thanks for coming. We appreciate it. And first off, we want to just start with kind of an introduction of just flat out what is LabArchives. So you may be familiar with LabArchives in working with other institutions, or maybe you have been using it for a little bit. We actually have 4 different products. We have the LabArchives notebook for research, which we're going to be focusing on today. We do have the notebook for education. We actually have 2 products called LabArchives Scheduler and LabArchives Inventory. I'm not going to cover the other 3 too much just because they are separate products and separate licensing. So we're going to be focusing on the ELN today, or ELN stands for Electronic Lab Notebook. Sometimes people call it Electronic Research Notebook or even Digital Research Notebook. And all of our tools are designed to help you and your researchers, your colleagues, your collaborators adhere to institution policy, but also government funding agency policy. We've been doing a lot of work with NIH 2023, but other similar policies in place to help researchers meet those needs, but really scale with a group. Maybe you're brand new to Weill Cornell, you've got a pretty small lab, but expect to be hiring on and getting some new grads, students, postdocs, med students, and growing with your team. And LabArchives is really designed to kind of grow with you as time goes on. Now, you'll hear a lot of different terms used for LabArchives: ELN, ERN, DRN, whatever you want to call it. It's a place for you and your colleagues to securely store, search, and share laboratory and research data online. All of our tools are cloud-based, meaning you can access them from anywhere. You can be on campus, you can be off campus, working from home, and easily collaborate and communicate with your team. And that's really the name of the game. I think if 2020 taught us anything, it's that we need to be able to collaborate and see what's going on in real time with our colleagues. So we'll be talking about some of the collaboration pieces as well, including things like, kind of streamlining the organization and tracking of the work that you do. Now, the most popular topic over the last year has been NIH 2023, which actually goes into effect next week. The clock is really ticking on this. If you have not if you are an NIH-funded group and you have not looked into some of the rules and institution policies in regards to NIH 2023, I'd recommend reaching out to some of the folks on the line today or to some of the resources on campus regarding this. It boils down to a couple of different points, primarily that all researchers must have a data management sharing plan. We'll talk more about this in a bit, but if you have not looked into this and thought about how this may impact you, definitely recommend taking a look at the NIH website as well as reaching out to your local team. On our side, we do a lot of work with NIH 2023 because often the notebook is part of that same conversation. The data management sharing plan tells you how are you collecting, organizing, sharing that data, and the notebook is often where that is done. So we'll be talking about some of the things that we do. You may have seen announcements at our working group, our advisory group, and some additional work that we've been having going on the last couple of months in terms of expanding some of those workflows. We are sitting there saying, hey, what is a data management plan, or what is a data management and sharing plan? It's often a formal document that you and your team create that describes what you do. What types of information do you collect? How do you collect it? How do you name and organize that data? Who in your team has access to it? Can they edit the data? Can they share the data? How do you onboard employees or offboard when people leave? How do you change their access to that data? But also thinking in the long term, 2, 3, 5, 10 years from now, how would you look back at this work? How do you want to access it? can someone access and work with this data several years down the road? Now, a data management plan can be a formal document. So if you're part of NIH 2023, there are some excellent templates available on that side. But if you just have a kind of informal agreement, and that's what a lot of groups have, just sort of, hey, well, we collect data like this, and we name it this way, and we put it here, and here's who we give access to. Having that written down somewhere, might be a LabArchives notebook where it's written down, might be a DMP tool, might be other resources on campus, can be so valuable. Because when someone looks back at your work 2, 3, 5, 10 years from now, they need to be able to understand what you did, how you did it, where you did it, and so forth. Most importantly, they need to understand all of that without speaking to you. That's sometimes a hard test to pass. And one of the main goals of our institution and our team and all of us is to increase reproducibility, to prove you are doing the work that you're doing and helping you to document that in such a way that you can prove that you are doing the authentic research that we all are doing as part of our workflows. As for creating an account, it is very easy. Go to labarchives.com. You will see an option to sign in through your institution, and Weill Cornell is on that drop-down list. You'll log in with your Weill Cornell credentials and get right on into your account. Let's talk about support. So you're very fortunate to have an excellent team on campus. Michele and Sarah are here from that team, and there are a few others as well. I believe they have some contact information and a website. They may send that in a minute. But you also have our support team. Our support team is available 8 a.m. to 8 p.m. Monday through Friday. Call, live chat, email. Please do get in contact with us if you've got questions, feedback, suggestions, want to dig in a little bit more. And we also have a knowledge base on everything LabArchives. Even though we have an hour today, I won't have a chance to cover everything. But we have webinars, which are publicly available. Anyone can register for. We have a YouTube channel. We even have quick start guides and more available for you all to learn more about different tools and features. So do feel free to reach out to some of those resources. And of course, the folks online today who are from Weill Cornell are great resources to reach out to on guidance, best practices, recommendations, and kind of building that community. The best person to reach out to for advice, someone beside you, someone at the institution. So there may be someone at Weill Cornell who could say, hey, I've done that, and here's what I did, or could give that kind of more hands-on feedback and advice. But let's talk about data. In research, data comes from all sorts of different sources, whether that is from equipment, whether it's observations and notes, and handwriting, maybe it's email, maybe you have pictures or videos or audio recordings, or maybe you even use some of the tools you see at the top there. Maybe you use Redcap, maybe you use Snapgene or GraphPad Prism or Jupyter notebooks. And all this data can be included into a LabArchives notebook. All these attachments, files, notes, observations, you can see in real time, you can share amongst your team and update, see what's going on, and you can also export. We provide several export formats for you all. We'll go into more detail on this towards the end of the presentation today, but you can always get the data back out. In good practice, in reproducibility, and good science, one of the most important things is making sure that down the road, you can get that information back out of your system. We're not going to manipulate or change your data in any way. It is still your files. It is still your research. It is Weill Cornell research. We're not going to, make changes to that. You can get it back out. You can work with it as you need, when you need. And let's talk a bit about permissions. Now, in LabArchives, central object here is the notebook. Every single notebook has a notebook owner. This person at the top, I almost always recommend it's someone like a PI, a department head, supervisor, team leader, because this person has full control over who can access the data. Now, often that notebook owner is someone like a PI. They're very busy people, and they may not be in the notebook every day. So they might ask someone else. Maybe it's yourself on the call today. Maybe it's a colleague in your team to have the role of administrator. Administrators can add, edit, work within the data, and invite other people. So they can actually go and say, hey, we've got a new grad student, postdoc, med student in our team. Let me go and add them. And that person typically has the role of user. User is sort of your typical colleague. Most of you on the line today will be a user in the notebook. So you can add, edit, and work within the notebooks. We do have this role of guest. This might be for undergraduate students, interns, volunteers, people outside of Weill Cornell who you collaborate could be guests. Guests have temporary edit access for up to 60 days. And after 60 days, it's view only. So often it's for kind of that short-term collaboration, someone outside of Weill Cornell, interns, volunteers, folks like that. The great thing is you can change roles. You can transfer ownership. You can adjust the permissions as you need. So you might set up the notebooks today, change your mind a couple weeks, couple months down the line. That's perfectly fine. You can always adjust that as you go. But just to show...
Anna, if I can
Yes.
interrupt a second, what we suggest is that the individual members in the lab can set up their own notebook, but then they transfer the ownership to the PI for several reasons. A lab member can leave, or also the PI is legally responsible for maintaining the data. So the best thing for us is for the individual member, because the PI might be too busy to create notebooks for everybody. So individual member in the lab can create their own notebook, but then they transfer the ownership to the PI. Thanks.
Sounds good. And just kind of expand upon that. As you mentioned, we typically recommend notebooks are owned by a central person. And this is an example diagram of what I typically see for a group. So maybe you have one notebook for shared resources or group resources. Maybe this is common lab protocols, policies, training for new employees, equipment maintenance. Maybe you have a weekly lab meeting and just the notes for the weekly lab meeting could be in one central place. Everyone in the team gets access to it, can add to it, edit, work within there. But a lot of groups start out with that individual notebooks. This is what Michele was describing. Each person of the team has their own notebook where they can add, edit, work. It's kind of their own place. They're sort of a professional portfolio. You know, day one of me at Weill Cornell, all the way to graduation day, all the way at the end of my program, in one organized place. But for a lot of groups, they'll move to a more workflow-based structure because the real benefit of scientific research, especially in the modern day, it's very rare that it's just one person working on something. Usually, maybe one person is out in the field collecting a data set. This person here is running an experiment based on that collection. And then this person down here might be analyzing that data, writing a publication. Maybe this person's an auditor who's checking in on the work. So you can have multiple people involved in that process, kind of that handoff as a project progresses, as a sample goes through a certain workflow, and you can all work together within the same notebook, within the same data, and see what's going on in real time. Now, these three are suggestions for how you might set up your notebooks. You might choose one of these, you might choose none of these, you might find a combination. But the best thing is LabArchives is very flexible. You can set up the notebooks in whatever way makes sense to you. You can always rename, repurpose, move around the data as you go. So it's very flexible on that front. So don't feel like you have to make a decision today on how you want to set up the notebooks. You can always change it down the road and discuss it more with your team. Now, I'm going to skip this slide and just talk about some of the benefits that are available. So for one, for your institution, it is protecting your intellectual property. There is a whole laundry list of server compliances, security, and encryption, and all that fun stuff that we do, and long acronyms of government funding agency compliances and other alliances, certifications, things that we do to help make sure that the work you're doing is secure, protected, and all that great stuff. But also for you as a researcher, LabArchives has a full revision history and audit trail. We'll go into more detail on this in a bit, but it allows you to prove that you did what you say you did. You can easily collaborate with your team. You can search across your notebooks. You can meet those unique workflow needs because often in science, every day is a little bit different. And it helps you to get started in minutes. So we'll show kind of today pretty much everything in LabArchives notebook, and you'll be up and running by the end. So if you have any additional questions, of course, our support team is here to support you. But it's not anything that's terribly complex or requires intense amounts of training. It's pretty easy. It's a lot like your paper notebook. You write your notes, you upload your attachments, and you're off to the races. Now, I want to get into a proper demonstration on how to actually use LabArchives. Before I do that, do y'all have any questions on anything so far? All right, I'm not seeing any, so we'll keep moving along. All right. So what you're looking at here is LabArchives. So I went to mynotebook.labarchives.com and created an account. I already have one, so I just logged in. And the center of the screen is what I call the workspace, or the page, where I can add, edit entries. The left-hand side is the structure of my notebook. Now we're in a notebook called Hannah's Notebook 2023, and it has some folders here. Maybe a folder for templates that I've set up. Maybe a folder for projects that are in progress. And on a page here, I can see some entries. So on my page, I might have some notes, some observations that I've made. I might have some items that I've used. I may have some results, various attachments that I've uploaded to this particular page. So I have an Office document, image, Jupyter Notebook files. If I use Jupyter Notebook, and even maybe some SnapGene files. So if you're someone who works with a lot of SnapGene or genetics file types, you can actually view those, see some details, the eye icon. And I can add data to this page. I can edit data on this page if I'd like, or click the word new, or I can create new folders, create new pages, or copy information. Copying is where you start seeing groups save a lot of time with LabArchives. I'm running the exact same experiment today, but changing a few parameters or adjusting the concentration or a couple of small changes. Great. Copy what you did before, update what changed, and get back to doing more of your work. With LabArchives, you can really start saving a lot of time and spending less time documenting your work, increasing the quality of your documentation, and getting more work done, which is the goal for all of us. When I click Add New Page, I'm going to make a blank page for today. So we're going to call this our demo page. Click Add. Now this is going to create a blank page for me, and you'll see I can drop files here, and I'll make some new entries in just a second. But if I right click on any option on the left hand side, I can rename a page, copy a page, and you do see an option there to delete. An important thing to point out about LabArchives: entries, pages, and folders cannot be permanently deleted from the notebook. I can always go in and review the revision history and undelete items. All that would happen, move down here to our deleted items bucket, where I can always right click and undelete and bring that right back on in. So I'm going to go and bring this right back into my account if I ever need to. At the word notebooks here, I'm going to see the other notebooks that I have access to. So the ones in bold are notebooks that I own or I manage, and the notebooks owned by someone else will say owned by and that person's name. If I want to create new notebooks, I can do that at the plus icon. So maybe I'm starting a new project and I want to make a new notebook. I can go ahead and create that. If you are the PI, you should see all the notebooks where you are listed. It's in nice and bold. If you are working under someone or you have a PI, you should see owned by. It should say your PI's name. So just keep that in mind as in terms of policy. At the top here, you're going to see the option to search. Pretty straightforward. You can search your notebook. But one of the most important features in LabArchives is our advanced search tool. This allows you to search across every single notebook in your account, the different users, tags, date, chemical structure, to find that work you're looking for, whether it was 6 months or 6 years ago. You can search across your notebooks to find more details there. Now, there are a lot of different filters that you can apply here, and search is the most valuable thing because often we talk with research integrity and scientific integrity folks. The hard part is finding the data. Once you find it, you can usually say, yep, there's the work. There's the, data for figure 4 in this, publication that was made. But being able to find it can be tough, especially when you compare it to the old school paper notebooks where you go, I think Jim worked on that. I think that notebook's over here. And I think it was over here, but maybe it's halfway through. And yeah, you're just having trouble tracking down that paper notebook or finding the files or keeping track of the work that was done. Now, on the top here, there are a few other options, such as your name, where you can sign in or out of your account, the bell icon, where you have notifications, the eye, where you have all of our help resources, our knowledge base, our quick start guides, and more. Then we have Course Manager and the inbox, inventory. These are two different tools. I won't go into too much detail on them. You're welcome to check them out if you'd like. For inventory, it'll start a 60-day free trial. So if you're interested in playing around with that, you can. If you've got questions, reach out to our support team. At the triple dot menu here, we have our notebook settings, our user management, and more. So we'll go into more detail on this in just a bit. Now, at the word new here, I can go and add some entries to my notebook. So there are a lot of entry types in LabArchives, and I won't have a chance to cover all of them today, but I'm going to show some of the most common. feel free to make some notebooks, make some pages, play around. I promise you can't break it. So for today, I'm going to click the option for rich text and create a rich text entry. Rich text entries are where I would have notes, observations, tables, images. So I can say something like super cool research. Great. And I can format this. So I'm going to make it nice and large, and I'm going to make it orange. That's Weill Cornell. Why not? And I may even center the text here. Of course, there's more options here. I can do links and tags and images and so forth. But when I click save, this entry is added to my notebook. A few important things happen. First off, there's some tools here for actually editing the entry. And then on the right-hand side, there is a date and timestamp. These are made by the National Institute of Standards and Technology, and they cannot be tampered with in any way. So these date and timestamps from trusted global time source allow you to keep track of when this entry was added to the notebook, who added it to the notebook, and I can go back and review that history there. So I can go and see the different versions of the entry. And most importantly, entries cannot be permanently deleted from your notebook. So you always have the record, January 17th, 4:22 p.m. Hannah added this to the notebook. Now, if I want to add some other entries to my notebook, click the word new. Again, I can select another entry type. Let's say I want to add some attachments this time. Select attachments, and I grab some files from my computer. Now I can do up to 50 files at once. So it can actually be a lot of attachments that I add to my notebook very, very quickly. And maybe I want to add an image, an office document here. And a good practice, especially when we talk about good research and good work that's done, is to add a file naming convention with your team. This is something that might be included in a data management and sharing team plan. And you can have good descriptions for your files. You can include this description field to provide more details. Maybe the software or hardware used to create the file, or other details like that to help you keep track of the work that's created, who put it in the notebook, how it was created, and so forth. When I click save, the files will upload to LabArchives. Built into LabArchives are viewers for Office documents. You can annotate images, you can snap change, genetics file types, PDFs, and more. So you can actually work with the files once they're in your notebook. Now, you'll see when I hover my mouse between two entries, there's even a little insert flag. Maybe you want to add something in a really specific spot. I'll add a heading this time so I can say final results below and click save. Now the heading just helps to break up the page a little bit. So it makes it a little bit easier to look at, kind of makes it, breaks up the spacing. Now, sometimes when we talk about attachments, next question is, well, what if I have a lot of attachments? Or if I have network drives, or I have equipment generating files all the time, and I want to bring that data into LabArchives. Well, one tool to check out is a tool called Folder Monitor. I'm going to bring up a screenshot of what this looks like. So Folder Monitor is installed on your local computer. It's available for Mac and PC. At the triple dot menu, you got the downloads, you'll see it right here. This is a screenshot of what it looks like. Essentially, you set up a rule and you say, hey, I want these files on my local computer to go to this location in LabArchives. So it's going to watch that folder. As soon as it detects the file, it's going to push it into LabArchives. And it will push new files, existing files, and it can be used to upload a lot of data into your LabArchives notebook. A lot of folks who have equipment will use Folder Monitor as a way to just watch that folder, push the files in all day long, and get that data into your notebook so you can kind of wash up, go back to your desk, and your data's there for you to start working. In a similar vein, our tool called the Microsoft Office plugin adds a shortcut to Word, Excel, and PowerPoint. So this is kind of just a nice time save. In the desktop version of Word, Excel, and PowerPoint, when you go to File, there's an option for LabArchives. So you can open up a file, work as you normally do in the desktop version of Office, and save the file back to LabArchives. So you can keep working as you normally do, the file's saved back, and your colleagues immediately get access. You've got all the desktop tools within Office. We also integrate with the online version of Office, which we'll show in just a minute. But I want to pause before we switch on over talking about actually working with the entries once they're in your notebook to see if there are any questions. And there's a great question for the Weill Cornell team. What is the maximum file size allocated to each user at Weill Cornell? Is there a limit to the file size?
The total space, there is no limit. The file size, I believe, is 16 gigabytes. No, Anna? That's what we have, I think.
So from what I'm seeing on my screen, the maximum individual file size for your license is 4 gigabytes per file.
4 gigabytes, okay.
Yeah. Now that is a per file or per attachment limit. In some cases, there may be a storage limit for the total notebook. I always recommend less than 30,000 entries per notebook, approximately less than a terabyte. We always joke, we'll send you a free t-shirt if you ever get anywhere near that, because very few people ever do. Because in it, realistically, you would have multiple notebooks. The reason we have that recommendation is just because when you start reaching that number of entries per notebook, that's a really active notebook with a lot of entries, and it starts getting a little bit messy. So generally, have as many notebooks as you need, kind of roughly that 1 terabyte limit per notebook. Some people call that unlimited, but it depends on your definition. The main limit is that per file attachment limit. Very good question. Okay. When linking files from other software, what happens if in the future that software is no longer accessible? Expired license, discontinued, unsupported. Is the linked file at least maintained as an image as it would be in a hard copy notebook? A trivial example, would the Snapgene plasmid map disappear if the license for Snapgene was discontinued by the university? What about a big Prism file? Will it still be able to open the file and use the data? Excellent question. And good to see that you're thinking the long term. You'd be surprised how many researchers don't think that far out. So when the file is right here, I have uploaded this file to LabArchives. It is stored within LabArchives notebook. So it doesn't really matter about your local license, whether it's expired, discontinued, supported. We will continue to store that file, even if it's some weird proprietary file format or whatever it may be. We will continue to store that file. Now, whether you can actually use that file, whether that is useful, is a great question. So let's say you have some strange proprietary file format and the software you use expires, discontinues, whatever. That's where I would probably recommend that you use the most common file formats available to you. This can be tough depending on the applications that you have. It's part of good data management and good research to use common file formats as applicable. So text files, images, PDFs, Word, Excel, PowerPoint, things that will be good for a number of years. I'd recommend talking to your institution about that specifically. They may have good practice recommendations, especially for Snapgene and GraphPad Prism. The great thing is those are really common file formats like PZFX. GraphPad Prism is like GPD. They're pretty good. But make sure that you're exporting to common and standardized file formats. No matter what, we will keep the file stored in LabArchives, but how useful it is just depends on what file format you've got. And they might be able to point you to some resources locally who can offer some advice on that. There's another good question. This is more of a question regarding compliance to NIH requirements. When files are transferred to LabArchives retrospectively, the date and time will not be the actual date and time of the experiment. Will this still be acceptable in terms of IP? Is there any way to stamp the real date of the file creation? It's a good question. So the date and time stamp that we show, top right here, will always be the date and time that it was put into LabArchives. So this is our trusted global time source date and time stamp. So there are cases where you upload this file, you can actually see it right here. I put this file in today, January 17th, but the name of the file says December 11th, 2022. In general, that's considered okay. Talk to your regulatory bodies, talk to your librarians, talk to your resources locally as to what they require. In general, it's considered good practice to upload it to LabArchives or upload it to something with date and time stamping as soon as you can. There are instances where that's difficult, and that's where I would recommend that you put it in the file name, put it in the file description, file created on December 11th and uploaded on January 17th due to vacation. I was out for a couple of weeks for vacation. I forgot to upload it. So I just put a note in there. In general, that's going to tell the full story. And that's kind of what a lot of folks look for. It's just honesty and, hey, it was created this date. This is why it was put in. For some institutions, they have LabArchives beside them at the bench. Some labs will have in real time, they are putting this file into LabArchives. Sometimes it's a few days, a few weeks later. I'd recommend talking to the regulatory bodies in place for you. If you're looking at things like patents, talk to your attorneys. They will be able to point you in a direction to what makes sense for you. And I will say, if the Weill Cornell team has a recommendation on either of those topics, just let me know. All right. I don't see any more questions, so we'll keep moving on. So let's talk about what we can do with our entries once they're in LabArchives. So we've got some notes about some super cool research. We've got an office document here. If I hover my mouse over any of these entries, there's some tools. Pencil icon, I can edit the entry. Arrows, I can move it around. Then we have this comment bubble. Let's say I want to ask my colleague for some advice on this. I want them to work on this. I might say at Jane Doe later today. Now, when I mention Jane Doe, she's going to get a notification in LabArchives and in email letting her know that I've mentioned her. She can reply back to me, and we can have a conversation about this particular entry of this page, of this folder, of this notebook. It's a really great way to draw someone's attention to a specific portion of the notebook, ask for advice, ask for help, and you can have that kind of dialogue in line with what's going on. Now, one of the things our researchers love about comments is one of the only pieces of information that can be permanently removed. If I click this trash can icon, it's deleted. But the reason that's helpful is you can talk as you normally do. Joke around, use it like a Slack or a Teams or kind of that chat sort of thing. And when you want it to go away, the trash can icon. So it's not part of the permanent intellectual property that the rest of your notebook might be. If you want to have that kind of formal dialogue, do that in line. Give the feedback in line within the entries, within the office documents. Now, we'll talk more about our activity feed in just a second, but you'll see that comment has been made. I can go up to the bell icon, and here's where I would have a notification. Oh, someone have a question there?
I think we have a couple, Anna.
Okay.
Yeah, so there is one from Serena, a very good question. Serena is asking, this is more a question regarding compliance to NIH requirements. When files are transferred to LabArchives retrospectively, the date/time will not be the actual date of the experiment. Will this be still accessible in terms of intellectual property? Is there any way to stem the real date of the file creation?
Yep, so we did answer that one. Main method would be through description, things like that. But the date and time stamp that we have will always be the same.
Also, the file should keep the creation and modification date. That's in
it
the
depends on your
main page.
operating
Depends on
system
the
and how
type,
that's configured.
But then the next one is it acceptable to take a picture of handwritten notes, upload to LA immediately, and then annotate, transcribe at the later date? Yeah, definitely, yes.
I think this is fine, yeah.
Yeah, absolutely. It's very common that folks do that. If you want a fancier option, there are it's called OCR or optical character recognition software applications out there. Essentially, these are fancy scanners. Some of them can be built into like phones and tablets. So you take a picture of your handwritten notes. It will convert it into searchable text, usually a PDF. I've heard really great feedback on a thing called Rocket notebooks. They're really cheap as well, which is nice, or Rocket books. There's also like smart pen technologies, if that's of interest to you. So there's kind of higher tech options, but absolutely the easiest is just kind of take your handwritten notes, take a picture of them. If they get into LabArchives, then you have a record of the work that was done there. So you'll have a record of the work that was done. Yeah, it's very common that folks just take pictures. My favorite, it was a presentation by a researcher a few months back, and she showed her real lab notebook. And one of the pictures in the lab notebook was just a picture of her glove, because she took notes on the back of her glove and just took her phone, took a picture. And hey, it's not ideal to write your notes on a glove, but it happens. Sometimes that's the closest thing by. So, it works. at least got in there somehow. And then you don't have to worry about when you throw out the glove. At least you have some record that you wrote the notes down. All right. So I mentioned the activity feed here. So those comments here are going to give me notifications. I can get email notifications about that. It can be in the activity feed as well. I can open that up at the top here. So I don't have any notifications in this case. I've already cleared mine out for today. I've been really productive this afternoon. But maybe I'm a manager and I just want to check in. I'm at the beach hanging out on my phone, just checking to see what the lab's been working on. And I want to set some filters here to see what's going on, see what I've missed while I've been out of town. This gives me some filters and I can see that, 3 minutes ago, Hannah edited this entry. She added a comment. She was working in some office documents. I can click on this to see more details, set filters to really drill in and say, hey, what has this person been working on? What's the status of this project? Kind of staying up to date on what's going on within a team and what people have been working on across the group. And when I close, brought right back to my page. Now, on our entry toolbar here, there's some other tools. I'm going to skip over sharing an entry right now. We'll come back to it. But at the triple dot menu, I can copy an entry if I want to, and even build links or tags. So someone asking about linking earlier, what if you have data stored elsewhere? Maybe you have data in another part of your LabArchives notebook. Maybe you have data at a website. Maybe you have data Box, OneDrive, Google Drive, wherever that may be. You could link to or reference that data to kind of tie the pieces together. And I think that's really valuable because sometimes in scientific research, maybe you do big data. Maybe you are a sequencing or imaging person and you have excellent Weill Cornell provided resources for those attachments. Keep them where it works, but provide those links, provide those references so the future viewer of your work can say, oh, cool, here's Hannah's notes. Here's her kind of a summary of what was done. And here's the link to the, raw files that might be stored elsewhere, could be in, your EDC system, could be in a number of different places. Now, also under that triple dot menu is an option for tags. Now, tags are a way to describe your data or add metadata to your work. This is a controlled vocabulary that you and your colleagues will build throughout your notebooks. So here you see I have a CRISPR tag that someone else has used. I can make new tags if I want to. This helps improve search. Now you could spend a lot of time with your team developing metadata schema and having an entire, dictionary, a data dictionary of what you use, or it can be more informal. And, for my lab, it would have been informal. We add terms as we go and you know what? This is part of our CRISPR project. So I'm going to add that tag. So now if I search for the word CRISPR, I'm going to find everything related to our CRISPR project, including this entry and this entry. And sometimes that's helpful to say, hey, show me the most recent result in this project and the most recent result with this particular tag. And of course, tags are all indexed. It improves search, improves reproducibility. It's a good practice to have in your team to use tags. Now, for things like office documents, I can click on the pencil icon here. This is going to open up the document. In this case, in Excel Online, do the same thing with Word and PowerPoint documents. And I'm going to see my file. From here, I can make changes to my file. So we'll just type in some new observations, some new numbers here. You're going to see as I type, it's saving back to my LabArchives notebook. And this supports simultaneous editing as well. So you and your colleagues can be working in the same document at the same time, and everything saves back as you're working. So when I close, my page will refresh with a new version of that file. Now, we also have an image annotator. This is a fairly simple tool. Often, if you're someone who does a lot of work with, handwritten notes or scribbles, something like this might be helpful for you. So maybe I want to circle the arrow here or add a quick label. Make it nice and large, hyperthyroidism, our little label. I can save it back to my notebook. Now that it's saved, when I close out, I'm going to see my label and my circle. It's going to tell me the annotation that was made here. Well, let's say I look back at this and I say, I didn't do a very good job. Let's go back to the earlier version without that annotation. Under that triple dot menu, I see an option for view revisions. This is going to show me the history of this entry. So it originally was uploaded at 423. And now we have the version at 440. I can click on it to see let's see, that was made by Hannah. Here's her IP address, what changes that she made. And if I want to go back to the original, click revert, confirm, we're back to the original. So we don't have those annotations. You know what? I don't need it on my page. I put in the wrong image. I didn't mean to put it here. Let's go ahead and delete it. Click delete, confirm. It doesn't show up on my page anymore. But as you remember from earlier, data cannot be permanently deleted from the notebook. Entries cannot be permanently deleted. So if I go to page tools and go down to view revisions, I'm going to see the history of my page. So at 418, we added the page name, we added some attachments, we edited an office document. At 441, Hannah Clark, my IP address deleted, and attachment. If I click undelete, click OK, it's brought right back onto my page, and that history there is still maintained. So now my history shows uploaded, annotated, reverted, deleted, undeleted. I have the record of what was done there, and I can always go back in and review that information when I need to. Now, once we have our page, let's say we've finished off on the work that we're going to put on this particular page. One of the tools I may want to use is page signing. Now, page signing is often used for groups with patents, for groups with FDA 21 CFR Part 11 compliance. If you ever talk with an attorney, they might encourage you to use it. For a lot of groups, it's not something you need right now, and that's perfectly fine. You can always add it in at a later date. And page signing is accessible to page tools up here. Now, when you sign a page, it is permanently frozen. You can't add anything, you can't edit anything, you can't remove anything. And you'll see a signed page right here, signed by Hannah Clark, January 3rd, and I can't edit anything on this page. It's permanently frozen. But let's say, maybe I want a check and balance system here. Maybe I don't want to sign the page. I want someone to approve it. So that's what's called page signing with witness. And I've got an example here of that as well. So this page was signed by my colleague back on January 3rd, and they've asked me to be a witness, meaning I'm expected to read through their experiment. So I can see the chemical they're making, the formula, I can see their reaction mechanism, the chemical products and reactants, the ChemDraw files and more. If I think this looks good, I'll click witness and permanently lock this page. It's going to show both of our signatures. Page signing and witnessing actually comes from the old school paper notebook days where you actually walk over to your supervisor and ask them to read your work and sign their name at the bottom. Now, let's say I read through their work and they were having a rough day. It doesn't look how I want. They've got some typos. I can click reject and give them some feedback here. Hey, here's what you should do differently next time, or here's some changes to make. Here's some suggestions. And the page will be unlocked. They can keep working, make corrections, and move on. Now, with page signing and witnessing, it's something I always like to show folks because it may be something that would save you hours every week. It may not be something you need right now, but we're seeing it as becoming a more common requirement for different funding and government agency work. So if you need it in the future, we have workflows for it to help support you and the work that you do. So maybe something just to keep in mind down the road. If you apply for new funding, they may ask for you to incorporate it in your process. It's pretty easy to do as well. Now, we do have an entry type I want to talk about briefly, which are what we call widgets. Now, widgets are a very broad group of entry types. So these are calculators for molarity, for dilution, for other work that you might do. And we provide a lot just out of the box with LabArchives. But you can also make your own widget. So I've got one at the top here that was made by a hospital we worked with. So they set up a call with us. We met for about an hour and helped them, develop this widget. But what it started out with was a paper form. So they have a blood sample, they collect from a mouse, they do some stuff to it, they count the number of cells. It's not that hard. But the problem was everyone was documenting it differently. It didn't matter if you look at Jim, Jill, or Johnny's work. It was all inconsistent. They kept forgetting to put stuff in, and they'd rename things wrong or not have the same consistency. So they set up a widget in LabArchives. So with a widget, you can have things like a calendar you can pick from. You can have drop-down lists. You can have checkboxes. And you can do some simple math. Now, this is not the kind of math, it's the kind of math I could do on the scribble on the side sheet of paper or in my head if I really want to. But by having it done for me, so I can just kind of type in the numbers as I see fit, keep working as I go. Does the math for me, reduces the number of just kind of napkin math mistakes I might make. If you are interested in learning more about how to build widgets and templates, reach out to us. We'd be happy to meet with your team one-on-one, offer some guidance, offer some suggestions. Something like this takes, I don't know, an hour to make, but can really save your team a lot of time. If you are someone on the line today who knows HTML, you know JavaScript, if you have a programming background, there's some cool stuff you can do in terms of making fields required, doing some calculations, having fields expand and collapse, some neat stuff there. So if you ever want to learn more about that, just feel free to reach out to us. We'd be happy to chat in more detail. Now I want to pause for a few seconds and see if there are any questions on anything so far before we talk about, user management, adding folks to the notebook.
No more questions at the moment.
Sounds good. All right. So let's talk about sharing your notebook. Most of the time, when you are sharing your notebook, it's someone who you work with day in and day out. Someone beside you at the bench, someone in your lab, in your facility. And if that's the case, go to the triple dot menu and go down to notebook settings. There are a number of settings you can adjust in terms of page signing and witnessing, font styles, and on user management, you can see who has access to your notebook. So I see I'm the owner, my other account's a user, someone else is a guest. And if I go to new user, I can type in someone's email address. If it's someone at Weill Cornell, you'd use their med.cornell.edu. If it's someone outside of Cornell, you'd use whatever email address that they prefer. One important thing to note, when you invite someone, they get an email letting them know, hey, you've been invited to this LabArchives notebook. They'll create an account and they will have access to everything on this left-hand side. So everything within Hannah's notebook. But keep in mind, that's all they have access to. I have other notebooks in my account. I haven't shared those with them. I could if I want to, but maybe those are private projects or maybe things that they're not working on, so they don't need access. I'm not going to share it with them. Under the role column here, I can adjust someone's role. So administrators can share the notebook with others. Users are kind of your long-term colleagues, whereas guests might be those undergraduate students, interns, folks like that. If you use page signing and witnessing, you can adjust those permissions here. With the trash can icon, you can remove someone. So Jane Doe has been an excellent grad student in our lab, but she's moved on. We're really happy for her. Trash can icon, click OK. Her access is removed. All the work, all the changes, everything she's done in LabArchives is maintained. All that I'm adjusting here is someone's access to the data. The other thing to point out on this page is the ownership transfer button right here. So maybe you're on the call today and you are planning to kind of set up the notebooks on behalf of your PI. Great, go for it. Make your notebooks, set them up, get everyone added. When you get a chance, transfer ownership to your PI, your supervisor, your manager, department head, whoever that may be. So that way you kind of know who owns those notebooks. Additionally, if you are leaving your position at Weill Cornell, be sure to transfer ownership of your notebooks to someone else continuing on with your work. Whether it's another PI, someone in your team, just go ahead and transfer ownership to them so they'll have access to that. Now, for the most part, when you're sharing, you'll use this tool. But sometimes when you're sharing, it's a small portion of the notebook. Maybe you want to give read-only access. And if that's the case, maybe you just want to share just one entry. Go to that share button. I can invite you to one entry if I want to. Or right-click on the left-hand side. There are share options here as well. Share. I can type in someone's email address. Here we go. I can give this person view or edit access. With view, they'll be able to view it. With edit access, they'll be able to edit. But I am only sharing this in progress folder. So when I click send, they're invited. They're going to have access to this particular portion of my notebook. There's even a little 2-person icon indicating that it's been shared. But when they log in, they're only going to see the information in the in progress folder. They have no idea about anything completed, anything else in the notebook. And again, they don't have access to my other notebooks, unless I choose to invite them, which I might. Now, the very last thing, I know we're not far off the hour, is how to export your notebooks. There are several different formats for that in LabArchives. If you go to the triple dot menu, go down to utilities, you'll see those here, create offline notebook and PDF. The PDF's kind of the most popular. Think of it like printing off your notebook, going over to the scanner, and just scanning each page. It gives you a table of contents, so I can see this is my notebook as of January 3rd of this year. I can click on the page and folder. I can read through what was done, see those date and time stamps, see those entries, and print this off if I want to. It's kind of the easy to use print off of my work. The other option here is the offline notebook. So it's going to look like this. It's a zip file, which would download to your local hard drive. It would be offline HTML, pretty easy to work with. It also contains any of your attachments that are under 250 megabytes. So you can go and take a look at that when you need. I know that's a lot of material for today, but I want to thank everyone for coming. We'll open up for questions. But if you have any questions after today, you are more than welcome to reach out to us. You can send us an email at support@labarchives.com. And if you go to the eye on the top right, you'll find our knowledge base, quick start guides, YouTube channel. Get in contact with us. We're happy to chat. There's a great question in chat. Can you expand on the integration with Redcap? Absolutely. So the Redcap integration is pretty new. It would need to first be configured by your Redcap administrators. So if you don't know who that is, reach out to whoever like the IT for Redcap might be. They will need to be part of the Redcap consortium, and they will need to configure that. There are instructions on how to do that in our knowledge base. So I'm pulling up the knowledge base article right here, and I'll send this in chat as well. So right here, you'll see that there is actually instructions for the Redcap admins, including a link out to a GitHub, which has some details on how to set that up, as well as there are some details in the Redcap consortium. So the Redcap repo, they should know what this means. If not, have them get in contact with us. We can look into it. In short, it allows you to, in Redcap, upload to LabArchives. You basically send a file from Redcap directly to your LabArchives notebook, and it's uploaded there. It's a pretty neat integration. It's a newer tool, but it has to be configured for each institution. And a good question, can this be used as an inventory management system for the lab? Yeah, it can be. So I will point out, we do have a product called LabArchives Inventory. If you want to check it out, you are welcome to. It's at the test tube icon right here. It has a free trial if you want to play around with it, but it is a separate product and a separate license. I won't go into too much detail on it today. If you've got questions, chat with you about it. But before Inventory came out, Inventory is only a couple of years old at this point. Folks did use LabArchives Notebook for inventory. There's a few ways to do that. I'd be happy to go into more detail and offer some guidance on that one-on-one if you'd like. Typically, it's done with an Office document, an Excel document, something like that, or even using like maybe each page represents a sample. So each sample is a page in the notebook representing all the attachments, files, images for that sample. And then maybe the notebook structure represents what freezer, what rack, what location it might be in.
Anna, can you show the widget for the freezer boxes? That's very useful, I think.
Oh, yeah. Not too frequently used these days. Yeah, so we do have a freezer box widget. I'll go and grab that. So this is a widget right here, which allows you to have a freezer box. So if you're not familiar, freezer box is a little grid that you can have different samples in. And let's say in cell A1, I have a cell line called Hannah's cells. I can fill out some details here. Now we give some default fields that were basically generated from interviews with our current users. Now you can go and add the different items here. Cell C1. We have a couple of these. Maybe we have a whole aliquot here. Yeah, we got a whole row here of Hannah's cells. And this can be a nice way to just put in the details into your cells, export to CSV, and display that freezer box on your page. Additionally, you can go the word new here. There's an option to actually import a CSV file so it can actually create that freezer box or database widget if you want to use that to the page. So this allows you to customize the different values that are used. So maybe you want concentration, maybe you want molarity, maybe you want pH. You can customize which values are used for each cell.
And just as a reminder to people, everything you put in one of these little cells is searchable. So you search for antibody against PIC or something like that, you can just search it and it's going to tell you where in the box is.
Yep. If you search the notebook, you'll find the various entries where it may be. Can Excel files be imported as well in the freezer box widget? Yes. So if you use that import CSV file, you'll upload an Excel file, a CSV file. There are some rules about how that's formatted. Go and grab. Our knowledge base has a sample file for you that is already pre-formatted, so I'd recommend using that one. Here we go. So here's what the file would look like. So you would have column and row information. In this case, I have primers, sequence, stock concentration, and notes about each sequence there. So I could then take this file and upload it right here. And in case you're curious, we do have the ability to display and work with freezer boxes in LabArchives inventory as well. So if you want to take a look at that, just let us know. I'm just not going into too much detail because it is a separate product. All right, well, we are close to time. I want to thank everybody for their time. I'll hang out for a few more minutes for questions, but it does look like they're slowing down a little bit.
Yeah, while we're waiting for questions, for anybody who signed in late, I just wanted to give a reminder that the session is being recorded. So probably likely tomorrow morning, you'll be getting a follow-up email from me with information, with the video link, as well as information about LabArchives that you can look up to help you as you're using the tool. Oh, you did just get a question. Can you export individual pages?
Yes. So if I just wanted one page here, or maybe my introduction page, there's an option right here for PDF. And that will make a PDF of just one page or just one folder. Yeah.
Any other questions before we wrap up? Michele, Sarah, was there anything that you wanted to say regarding LabArchives before we finish up?
I just wanted to add one thing. So maybe some of you are still using paper-based lab notebooks, and we strongly encourage you to switch to LabArchive. This will highly simplify your life to comply with the NIH new data management and sharing policy, and also with the Cornell University data management and retention policy. We're happy to provide any one-on-one any kind of consultation to help you switch to LabArchive if you need, or if you need a specific training on that or specific help with that, please do not hesitate to reach out to me or Michele. But yeah, think about transitioning to LabArchives as soon as you can, and drop the paper-based lab notebook as soon as you can. That's all I had. Thanks a lot, everyone.
Thank you, everyone. Thank you, Hannah, so much for this great session. We really appreciate it.
Happy to be here. Thank y'all so much. You have a great afternoon. If you need anything from us, please reach out.
Awesome. Thanks, everyone. Have a good evening.